entitled Advantages in Automated and Digital Tissue Engineering and 3D Cell Cultivation. The topic will be delivered by Dr. Manfred Taschner, who is Chief Executive Officer of LifeTech, where he focuses on solving the most critical challenges of cell cultivation. He has long experience in preclinical field, and then he moved to with the decision to build a company exactly targeting this crucial step during drug development by simulating the human body in a medium to high throughput scale for automated drug testing in order to get essential information about the potential effect of drugs in the human or animal body without the need of introducing the substance to the body. So thank you very much for joining us, and I'm handing over to you.
Yeah, thank you very much for the introduction. You basically summarized everything I'm about to say already. Nevertheless, start my presentation now. All right, so thank you very much for inviting me to give a talk about the advantages of the automated and digital 3D cell cultivation. You basically said already everything about me in person, but maybe just to give you an idea of how I got into this whole topic, I will just talk a few words about me first. So I studied biotechnology in Vienna, then went abroad to Australia and finished my studies at the Harvard Medical School in Boston, Massachusetts. After coming back to Austria, I started, as you already mentioned, at a pharmaceutical company. They were focusing on the development of drugs or vaccines against Alzheimer's and Parkinson's disease, had already more than 100 employees and were funded with several hundreds of millions euros. But in the clinical phase 2, the results suggested that the placebo was actually working much better than the vaccine itself. This was, of course, a very disastrous result. And so I, myself and my co-founder, Volker Lorber, we both brainstormed a bit how we can really improve this whole development phase and can really have an increased success rate during drug development. But in order to really go into detail, we must first look at the drug development process in general. I guess you're all familiar with that process. So you're basically coming from basic research to drug discovery, where you have several thousand substances that are potentially a drug candidate. As soon as you have first testings, you go into the preclinical phase. This is where my company is now focusing on, where you test with traditional 2D in vitro cell cultures and also in vivo animal models in order to get an idea of the efficacy and the toxicity of your potential drug. Afterwards, you enter the clinical phase with only a small amount of substances, and I guess we are all aware of these phases. Eventually, you will have one medicine product in the end. So there is still, as I said, a high potential of increasing success rate during drug development. I took this beautiful picture here from a publication from Gail van Norman. You see, it's only 1 out of 10 drug candidates pass the preclinical trials, and that doesn't mean that the drug is already about to hit the market, because again, only 1 out of 10 of these drug candidates that passed the preclinical trials and now entered the clinical trials pass these further trials. So devastating success rates. This is the biggest issue is here the poor correlation of toxicity and efficacy of animals in comparison with humans. Since we are not rats or mice, but humans, and we develop drugs for the human body and not for any animals, there's a major potential behind improving this whole process. Yeah, 50% of all drug failures in clinic are due to the toxicity issues in the human body, meaning a mouse or a rat can easily have no problems with a certain substance, but in humans, this is a whole different organism. It has different immune systems, could lead to severe problems during clinical phases. And also, of course, the costs of in vivo trials, speaking of animal models here in the preclinical phase, is about 30% higher than in vitro models. So this is also something where there's a lot of potential that you can save money during the drug development in general. To summarize the problems in the preclinical research, let's start with the very poor standardization and reproducibility. It's kind of a very strange thing that in 2021, people are still doing most of the biotechnology research manually by hand. We all are aware of digitalization taking over. We all have our smartphones, tablets, and other fancy devices. But as soon as we enter a biotech lab, we put them aside and use manual pipettes to go back more or less into the dark ages of efficiency. The same is the same problem also occurs. I mean, this is also a consequence of the manual approach, that we have high financial and time requirements in general for the drug development process. So we're talking about more than a decade for a drug normally. Of course, there are COVID ways to really increase or accelerate the whole process. But in general, we are talking about several hundreds of millions euros, or maybe even $1 billion, and more than a decade of time requirements to get a drug to the market. Yeah, in terms of the prediction of the drug efficacy and also toxicology and toxicity, of course, in humans, we have very little to almost no prediction in this preclinical phase. As I already mentioned, rats are not the target organism, so it's great to have a potential drug, for example, for Alzheimer's that works in rats, but you won't really make a lot of money with that, and you won't get people to buy such a product if it doesn't work in humans. And these days, it's also very important to keep the ethical questions behind the in vivo experiments in animals in mind. The European Union recently, well, it's a couple of years ago already, provided us with the 3 Rs, the reduction, the refinement, and the replacement of animal models. But in reality, animal models are still the gold standard, and the FDA and the EMA are still require data from animal studies in order to give green light for the next phases. Now let's take a look maybe also at the in vitro cultivations that could replace or reduce at least the animal models, where also human cells can get involved to give a better prediction of future clinical trials. I took this beautiful picture here from a different publication from Sigrid Langhans. I think it's a nice overview of the different approaches. So we have this traditional 2D approach where the cells are growing on a plastic surface. In terms of physiological environment, this is of course not really natural. I haven't seen any people growing on plastic surfaces so far. And it's also, of course, causing problems for morphology and really physiological environments for the cells, and they change, thus giving different results than in vivo experiments. On the other hand, we have 2.5D cultures. These are cells, they grow on a coated surface. Most of the time it's collagen 1 or similar matrix. And still, it's only one layer of cells. It's physiologically more relevant, but still it's not an in vivo state as we need it for a really good prediction of the efficacy of drugs in the human body. So we really have to focus on 3D cell culture to get a better impression of the efficacy. And there are several different approaches. I want to start with the non-scaffold-based models. Most of them are really going into directions of spheroid and organoid cultures. I guess you're familiar with these methods. They have the hanging drops where the cells are building these spheres. The same is true for the low attachment plates where the spheres cannot attach to any plastic surface and they are more floating, but really floating is only the magnetic levitation approach where the cells are linked to magnetic beads and can therefore have access to media and nutrients all over the sphere and do not attach to the surface. Before we go to the scaffold-based ones, maybe let's take a look at the specialized 3D cell culture platforms. I guess you're also familiar with the microfluidic devices and micropattern plates. These are very detailed reproduction of whole tissues and even a whole human body where you have several compartments of tissues and organs that you can really test your drug candidate with. The only problem here is that it's quite an expensive approach and you can only test one specific sample at a time, and afterwards you have to get a new microfluidic device because the first experiment may interfere with future experiments. So for high throughput systems, this is not really something we can use. When we go to the scaffold-based approaches, this is much different there. We have, most of them are, they have hydrogels as a basic matrix. We have biological and synthetic hydrogels, giving us really an environment for the cells where they feel like in vivo. This is, of course, only simulating the in vivo status, so you will never really replace, well, from point of view now, you cannot really replace the in vivo testing, but you can at least get a good prediction of how the cells could react to your drug candidate in an in vivo state. We don't want to forget the newest technology on the market and very promising as well, the 3D bioprinting. So we have these great prototypes all over the world showing us hearts designed and printed on a computer more or less, also ears and other tissues, very promising, but the biggest problem here at the moment is really to get a functional organ. They really need nutrients, they really need vascularization to get a functional organ in the end, or tissue-like structure. What basically is the key, or what the people are really aiming for these days, is to also print these vascularization structures and get the organs to run. Most of the time they also cover it with some hydrogel to give this physiological environment again. It is a good approach. In my opinion, it will probably take another 10 to 15 years to really get it that much evolved that we have functional tissues to test our drugs with. Of course, this is also the first step towards automated cell cultivation as well in the three-dimensional environment. And I would like to show you how we are approaching this topic. So we built a robotic system where we have a completely new approach of the cell cultivation. We have a so-called vertical cell cultivation that was originally designed for automated cell cultivation. As you can see here, the cells are growing in these scaffold holders, as we call them. They can be further easily transferred from one media that is depleted to a new media, and then we can easily test the whole cultivation process, and also have precision approaches with syringes and also for media supply. This is the whole robot here in our new lab. This was the picture from May when we just moved into our new lab. So there's not much of the lab equipment set up at that point, but I think you get a basic idea of where we're heading. So really 3D scaffold-based cell cultures and tissue generation for an automated approach. So when we summarize the manual versus the automated cell cultivation, some of the very obvious flaws of the manual approach pop up. So as we already discussed, there is a very low reproducibility. I think we're all aware of the operator variation. That is a major problem in biotechnology. When you have two technicians, they perform the same experiments, you can be sure, even if they use the same SOP, they have two different results. This is due to small differences in handling samples, and also everyone is working differently at the bench. And automated approaches, of course, does this with always the same process. This is also true for standardization. Of course, you can have one technician who's doing all the work every day, the same thing, but still he will have variations in his results. And automate the robotic system has the possibility to really have the standardized product output in the end. When you handle your samples manually, there's a major risk of contamination, of course. Compared to a robotic system where everything is closed, there's no humans involved, no potential bacteria or viruses introduced into the system. Much more safe, of course. Yeah, we are all humans, we all do mistakes, and this is why the manual work is also prone to errors. Of course, the machines also do mistakes, but most of the time they can be traced back to humans, first of all, and then they can also be solved, and really in future processes they not occur anymore. So the goal and really the idea is to have always the same process going on. And this is also, of course, as I mentioned, it's already the time and cost consuming when you do everything by hand. You need a lot of workforce, a big workforce, several technicians doing it. And an automate doesn't really care about night shifts. He's working 24/7 and really get a much better output than manual approaches. That's much for the automated 3D cell cultivation. I also was talking about, or promised you to talk about, the digital approaches. Maybe just to summarize what this will be about. We have, first of all, the topic of laboratory information management systems, which is more or less the software behind, should be the software behind every lab, to really get a good overview of how the processes are going on and how the clients react to it, and also how your whole team is contributing to this research. Then I will talk a little bit about the industrial Internet of Things and digital twins in biotechnology. This is, of course, something pretty new, and not many people can think about applying this. And eventually, if the digital twins and Internet of Things approach works, you can also think about machine learning based in silico experiments, where you can really in the future potentially avoid animal testings and even in vitro testings at some point, when you have an experiment basically in a virtual environment performed and giving you an idea of the efficacy and the toxicity of your drug candidates. So let's start with the laboratory information management system. So as I said, this is a software sort of that is really covering all major parts of a company and potentially also a academic research lab. It starts with your clients and the communication with clients, that this is logging all the information that is needed, putting alerts and also analysis reports online. Then you have also your laboratory organization in the software, where you have access to your accounts, departments, and authorities to certain members, and stock management as well. You can create spreadsheets and can really even go for auto calculations where you have analysis done to a certain degree by your LIMS system. And this is, of course, important when you have several instruments that need to be connected and data needs to be imported and exported. And eventually, I can only tell you it's very important to have a good validated LIMS, because the EMA and the FDA will really look into that if that is approved at a certain degree. Yeah, we also have sample management. When I go back into my times from the university, I remember there was this lower drawer in the fridge or in the freezer where there are boxes in there when nobody knew which samples are stored there. This is all the past, because you now have a barcode system and you can exactly see where your samples are stored and what kind of experiments were performed with them. Yeah, I already mentioned that the import of data is very important, of course, because several instruments have different data formats. This is why you also need to have an export of data in this LIMS system. And there are several additional functions as well, customer or client-specific ones, where you can really go wild with your ideas of what to implement in these systems. Now let's talk a little bit about the digital twin approach for the automated cell cultivation. This is only possible due to the automated cell cultivation because you need a lot of sensors introduced into your robotic systems that can retrieve data from analysis results and also machine data and combine them to improve the system during its normal process. So the basics for the digital twin is really the industrial Internet of Things and the real-time data processing. You need a homogenized data set that needs to be decoupled from the physical experiment, saying you can simulate what might happen when you, for example, increase the temperature of your cell cultivation. And then with this information, you can really go back and reprogram the whole experiment in order to get better results and a better process going on. So this is why the physical object or the cell culture, when you're talking about biotechnology, should be accessible to alterations due to an artificial intelligence decision. Of course, there will always be potential problems or issues in a cell cultivation, because we're talking about biological data here that is from its nature very not constant, let's put it like that. And when you have an issue detected with your cell cultivation, you can always go back and trace back the problem to improve the process and avoid these issues and wrong tissue cultures, when you want, as you will, in the future. And eventually you need a modular approach where you can easily define, design, customize your whole experiment for better performance, meaning the machine learns how to improve the process and becomes better over time. As soon as you have something like this employed in your lab, you can go ahead to in silico experiments. I already mentioned this is kind of the future. It's not sure that we will go there soon, but the idea is that you have an algorithm.
Dr. Taschner, can you please wrap it up, because we have also questions from the audience?
Oh yeah, sure. Okay, it's the last slide anyway. We have artificial intelligence, more or less, using big data to predict future clinical and preclinical trials and suggesting ideas of which experiments are really necessary and which can be avoided. As promised, I'm done, and I'm open for questions. And if you need more information, you can always contact me via email or just call me.
Thank you very much. May we get a question from the audience?
Yes, the question is from one of our panelists, Dr. Simeonova. She is asking, what do you think? Will the results be comparable to the processes in the living organisms when you use 3D printed tissues for drug testing? Because this way you cannot recreate the body's immune response or changes in the microbiome, for example.
Thanks for that question. Yes. Yeah, as I said, this is the way we are heading right now. I don't think we can really get completely rid of in vivo trials, and especially in humans. This will always be part. It's just a good idea to give you an impression of the efficacy in humans. I think my second slide showed that the major problems are toxicity and efficacy issues during preclinical trials. And the idea is really to give our clients a better idea of which products are really very lucrative and can have a high success rate during the clinical phase, and others that can be easily discarded at the beginning because, yeah, it's just not really working in the human body.
Thank you.
Thank you very much. It